The cardiovascular system. The objectives of the class is the students should know about the pulmonary and systemic circulation, the portal system and various types of portal system, structure and function of the various blood and lymph vessels, and correlate the structure and function with some pathologic conditions. The heart acts as an efficient central pumping system which pumps the blood to the lung or the periphery. From the larger blood vessels, the blood goes through the smaller blood vessels into the capillaries. The arteries deliver blood to the capillaries, and two-directional exchange of fluid and gas occurs in the capillary bed. The arterioles, the smaller division of the artery, it regulates the blood flow through the capillaries. The microvascular bed consists of arterioles, capillary network, and post-capillary venules that forms the microvascular bed in the periphery or in the lung. So, there are two circulations, the pulmonary circulation from the heart to lung and back to heart. The systemic circulation from the heart to the tissues and back to the heart. In addition to the systemic and pulmonary circulation, there is portal circulation. The portal system consists of vein or arteriole interposed between two capillary systems. There are hepatic portal system, hypothalamo-hypophyseal portal system, and renal portal system. In the hepatic portal system, the capillaries are present in the intestine. They join to form intestinal veins. The intestinal vein joins and enters into the liver. In the liver, again it divides into sinusoids or capillaries. Those sinusoids join to form the hepatic vein, and the hepatic vein drains into the inferior vena cava. In case of the renal portal system, there is afferent arteriole which enters the glomerulus and divides into capillary network. Then capillary network counts as efferent arteriole. The efferent arteriole breaks into smaller capillaries in the collecting tubules of the kidney. Then they rejoin to form smaller tributaries of the renal veins. There is an efficient hypothalamo-hypophyseal portal system. The blood vessels from the hypothalamus, they divide and form a capillary plexus in the stalk of the pituitary gland. From there they join again to form bigger blood vessels. Those blood vessels again divide in the posterior lobe of the pituitary gland, form sinusoids. Those sinusoids join to form the smaller pituitary veins. So there are arterial portal system and venous portal system. Basically, either artery divides into capillaries or the The vein divides into capillaries and they rejoin to form bigger vein or bigger arteries. In portal system, there are two sets of capillaries. In the hepatic portal system, one set is present in the intestine, the other set is present in the liver. In the renal portal system, one set is present in the glomerulus, the other set is present in the collecting tubules. General features of the arteries and the veins. There are three layers from lumen onwards: the tunica intima, tunica media, and the tunica adventitia. The tunica intima consists of three parts: the endothelium, which is simple squamous epithelium; subendothelial connective tissue. Just below the endothelium, there is some collagen fibers and reticular fibers; the internal elastic lamina. So, the tunica intima has three components: the endothelium, subendothelial connective tissue, and the internal elastic lamina. The tunica media is the thickest layer in bigger arteries, the muscular arteries, elastic arteries, and also in the veins. Lymph vessels also contain, bigger lymph vessels also contain some amount of muscle, smooth muscle in the tunica media. The tunica media extends from the internal elastic lamina up to the external elastic lamina, and it consists of elastin, smooth muscles, reticular fibers, proteoglycans. The contraction of the smooth muscle cells regulate the blood flow and blood pressure. There are two terms: vasoconstriction and vasodilatation. Vasoconstriction is increased vascular resistance, and that is maintained by the tone of the smooth muscles of the tunica media. Vasodilatation or decreased vascular resistance is produced by endothelial derived relaxing factors and nitric oxide. The tunica adventitia contain longitudinally arranged reticular fibers, collagen fibers, and some blood vessels and nerves. Those blood vessels supplying another blood vessel are called the vasa vasorum, and the nerves are called nervi vascularis. They are mainly the cholinergic and the adrenergic nerve fibers. Arteries have been classified into three types on the basis of the size and characteristics of tunica media. The large arteries, most of the large arteries are elastic arteries, while the tunica media consists of predominantly elastic tissue and less muscle fibers. Examples are aorta, pulmonary trunk, brachiocephalic trunk, common carotid artery, subclavian arteries. The cross-sectional diameter of the ascending aorta and pulmonary trunk are each about 30 millimeters. Functions: they act as reservoir of blood which is ejected from the heart by expansion of the arterial wall. By the elastic recoil, the arteries convert the intermittent flow of blood from the heart into a constant one or the continuous one. Elastic recoil of the arteries maintains diastolic blood pressure and helps in apposition of the aortic and pulmonary curves during diastole. The coronary circulation is increased in diastole due to same reason. The muscular arteries. Most of the distributing arteries are muscular arteries, where tunica media consists of more muscle fibers and less elastic fibers. The muscles are mainly composed of circularly arranged smooth muscles which can respond to nerve stimuli and regulate the size of the lumen of the distributing arteries. The small arteries or arterioles. They are the smallest divisions of the muscular arteries possessing three coats. The cross-sectional diameter of the arteriole is about 100 microns or less. As the arteriole progressively divide into smaller branches, their coats become thinner and successively terminal arterioles or meta-arterioles are formed. Here are some more details about the elastic artery. The walls of the elastic artery have multiple sheets of elastic fibers in the media. The elastic recoil maintains the diastolic pressure and makes the intermittent flow to continuous flow because of the elastic recoil. The tunica media contains smooth muscles which are separated by fenestrated elastic fibers, and through these fenestrations only the nutrition from the lumen pass to the wall of the vessels. At birth, the elastic lamina are absent or very few. In adults, there are 40 to 60 lamina, and in hypertension and old age, elastic lamina increase in thickness and number. The smooth muscle cells synthesize collagen, elastin, extracellular substance, platelet derived growth factor, fibroblastic growth factor, which are required for maintenance of the tunica media. The adventitia is relatively thin and contain connective tissue, collagen fibers, some elastic fiber, fibroblasts, macrophages, blood vessels that reach up to the middle layer of tunica media. The inner part of the tunica media gets nutrition from the luminal blood vessels, luminal blood by diffusion. The endothelium contains Weber palate bodies that contain von Willebrand's factor. The endothelial change in response to various stimulation called endothelial activation. The inducers of the endothelial activation are bacterial, viral antigens, cytotoxins, complement producers, lipid products, and hypoxia. This is hematoxylin eosin staining of the wall of a elastic artery. The upper half shows the low power magnification and the lower part the high power magnification of the tunica media. In the tunica media, there are muscle cells, smooth muscle cells, where you can see the nuclei, and in between these wavy areas are the elastic lamina. Elastin doesn't take the uh, proper stain. With using it gives a fluorescence appearance. When you see under the microscope, you can see the fluorescence and can identify the internal elastic lamina, external elastic lamina, and the elastic fibers in the tunica medium. To see the elastic fibers, a special stain is done that is called wear stain, where the fenestrations and the undulations of the elastic fibers are visible. The endothelium of the blood vessels are very important. The endothelium is simple layer of squamous epithelium, and the long axis is of the endothelium is along the long axis of the vessel. And in scanning electron microscopy, the position of the nucleus or the elevation of the nucleus is also visible. This picture represents the two endothelial cells. They are connected by junctional complex, and these junctional complex are very important for functions. On the surface of the endothelium or the luminal surface contains certain villi, receptors, cell adhesive molecules, and some of the granules, Weber palate body which secretes anticoagulants. All three kinds of junctional complexes are seen, and during inflammation what happens, especially in the postcapillary venules, they allow substances from the lumen of the blood vessels to outside to produce inflammation. Properties of the endothelial cells. The endothelial cells are very important. They maintain a barrier between the luminal blood and the extracellular connective tissue. From the lumen, certain substances are diffused or they are actively transported, or the endothelial cells engulf something by pinocytosis, or they are stimulated by certain chemicals where the receptors are present on the endothelial cells. Like for example, the simple diffusion of carbon dioxide and oxygen occurs at the lung. Then there is active transport of glucose, amino acid, electrolytes, water, and small molecules, LDL, cholesterol, transperatin, growth factors through the endothelial cells. They also maintain a non-thrombogenic barrier. They secrete anticoagulant, antithrombogenic agents, and prothrombogenic agents also. They secrete vasoconstrictors and vasodilators also. They also regulate certain growth stimulating factor, and they also secrete certain growth inhibiting factors. It regulates lymphocyte migration by expression of cell adhesion molecules, and they regulate the immune functions also. They synthesize basal lamina and glycopollax, and they produce free radicals, and they oxidize LDL. So The endothelium of the blood vessels are very important functionally and maintenance of the normal milieu. This is one example what happens if there is slight disturbance in the endothelial function. There is expression of cell adhesion molecules by the endothelial cells and the monocytes are attracted. Monocytes pass through the gap between the endothelial cells and they uh, contain or imbibe the LDL, which looks like foams. Those are called foam cells also. These endothelial cells secrete PGDF, which stimulates secretion of cytokines and interleukins, and they stimulate the proliferation of the smooth muscles. And because of the proliferation of the smooth muscles and the foam cells, the, the there may be elevation of areas inside the lumen, and because of the friction by the flowing blood, the platelets get adhered to these areas and they form thrombus. The thrombus becomes larger and they break down to form emboli, and the emboli may lodge into various smaller blood vessels, and if the emboli are lodged in the blood vessels of the heart, it can produce angina or myocardial infarction. If those embolus goes to the smaller blood vessels of the brain, it may produce stroke. Similarly, there may be deposition of cholesterol in the tunica media, and that may close or constrict or narrow the lumen of the blood vessels. As a result, there will be less blood supply to the area supplied by these kinds of blood vessels. Normally, uh, the, the cardiac blood vessels are blocked by these kinds of atheromatous plaques. Already I have mentioned about the muscular arteries. The tunica media of the muscular artery contains large number of smooth muscles. In between there are elastic fibers are there, and these elastic fibers are secreted by the smooth muscle cells. Some collagens and some reticular fibers are also synthesized by the tunica media cells. In muscular arteries also, you can see the tunica intima containing all three layers, that is the endothelium, the subendothelial connective tissue, and the internal elastic lamina. Outside there is external elastic lamina, and the tunica adventitia contains connective tissue, collagen fibers, elastic fibers, and some blood vessels and nerve fibers. Muscular arteries have almost entire thickness of smooth muscle and less elastin in the tunica media than the elastic arteries. The tunica intima is thinner and contains prominent internal elastic lamina. The tunica adventitia is relatively thick, often separated by external elastic lamina. This is the picture of the or comparative picture of the elastic artery and the muscular artery. One can see the all three layers: intima, the media, and the adventitia. The tunica media contains wavy elastic fibers, and in between the elastic fibers are the smooth muscles. In case of the muscular artery, internal elastic lamina is very prominent because after death there is contraction of the muscle fibers. As a result, the elastic fibers becomes wavy. Under the microscope with hematoxylin and eosin stain, this layer is a refractile layer. So that is the one of the identifying points of the muscular artery. In case of the elastic artery, internal elastic lamina is there, but it is mixed up with the tunica media fibers, and contraction of these elastic fibers are not so prominent than the muscle fibers. It is because of the contraction of the muscle fibers of the muscular artery, the internal elastic lamina becomes wavy. Adventitia contains the connective tissue in both the both type of arteries. The muscular arteries divide and subdivide and form the small arteries and arterioles. All three layers of the of a blood vessel are visible in the small arteries and arterioles. The small arteries, the tunica media contains uh, up to 8 to 10 layers of smooth muscle, and in case of the arterioles, the tunica media consists of hardly 1 or 2 layers of smooth muscles. Here is the difference between arteriole and small artery. The diameter of the arteriole is 10 to 100 micrometer, and it contains 1 to 2 layers of smooth muscle. Internal elastic lamina is absent or poorly defined in the arteriole. The tunica adventitia is ill-defined connective tissue. In case of the small artery, the diameter is 0.1 to 2 millimeter. The size is bigger, and the tunica media contains smooth muscles up to 8 concentric layers. The internal elastic lamina is present and prominent. So the difference between the small artery and arteriole is in the internal elastic lamina. In case of the arteriole, internal elastic lamina is absent or very poor, but in case of the small artery, internal elastic lamina is present and prominent. The tunica adventitia is well defined in case of the small artery. Here is an example of small muscular artery. One can see thick layer of muscles, smooth muscles in the tunica media, tunica adventitia is there, and this wavy appearance towards the lumen is the internal elastic lamina. Because of the contraction of the smooth muscle after death, this internal elastic lamina becomes wavy. So, to identify a small artery, you have to see the internal elastic lamina. See here also, this is one of the longitudinal section of small artery, and the larger arteriole. Actually, there is no demarcation between these two. So one can see the thick layer of muscles, the endothelium, and the internal elastic lamina. The function of the arterioles are to regulate blood flow in the next subdivision of the blood vessel, that is the capillary bed, and they increase the vascular resistance, and they form the precapillary sphincter. So these smooth muscles acts as a sphincter and regulate the blood vessel, regulate the blood flow into the capillaries. Again, there are examples of arteriole. Here is the lumen of the arteriole, and the wall contains cross section of the smooth muscles. There is another one here. You can see the wavy appearance and more than 2 layers of smooth muscle cells are there. So this is also one of the arteriole. How to distinguish between the arteriole and venule? The venule has smooth muscle wall also, but the lumen is larger than arteriole, and the wall contains few layers of muscle cells, smooth muscle cells. This is one of the venule. It is containing the RBCs inside. And how to differentiate between the venule and lymph capillaries? There are RBCs inside the venule. The lymph capillaries are bigger, irregular, and they do not contain RBCs. The next subdivision of the blood vessel or after arteriole is the capillary. So these three pictures are different types of capillaries. See, the most prominent feature of the capillary is that absence of smooth muscle in the tunica media. The capillaries has only endothelium. In electron microscopic picture, one can see the nucleus of the endothelium, and another cell is there just outside the endothelium, is called pericyte. There are small gaps are there and bigger gaps are there. Where bigger gaps are there between the adjacent endothelial cells, these are called sinusoids. So there are three types of capillaries are there. Those are the continuous capillaries. This is the capillary endothelium lying on the basement membrane, and two endothelial cells are joined by junctional complexes. Pinocytic vesicles are visible. This type of capillaries are present in the brain, continuous capillaries. They don't allow the substances from the lumen to the substance of the brain. Fenestrated capillaries. There are gaps in the endothelium. In the cytoplasm of the endothelium, there are certain areas where the plasma membrane are very close. Junctional complexes are there, and these gaps are lined by diaphragms. Basement membrane is there. So this kind of capillaries are present in the kidney, glomerulus of the kidney, and these gaps Through these gaps, substances pass from the lumen of the capillaries to the Bowman's capsule or the urinary hole of the nephron. And discontinuous capillary or sinusoids. There are big gaps between the endothelial cells, and there are gaps in the basement membrane also. So this kind of capillaries are present in the liver. Again, there are examples of three types of capillaries: the continuous capillaries, the fenestrated capillaries, and the sinusoids. Here are the examples of the continuous capillaries. So this black line is representing the basement membrane, and this bluish area is representing the cytoplasm of the endothelium. In case of the fenestrated capillaries, the plasma membrane of the apical side and the basement membrane comes together, and there is a diaphragm. Through this diaphragm, or the diaphragm acts like a filter paper. Continuous capillaries are present in the brain, and the fenestrated capillaries are present in the kidney. See the appearance of the capillaries. The capillary's diameter is maximum 5 to 7 micron, and inside the muscle one can see the RBCs inside the capillaries. The normal diameter of the RBCs is 7 micrometer, so the diameter of the capillaries is just enough to allow the RBCs to pass from one side to the other side. The microcirculation or arteriovenous shunts. So till now we came to know the larger blood vessels divides into smaller blood vessels, then the smaller blood vessels divide and subdivide to form the arterioles, and after arteriole there are blood vessels called meta-arteriole. The arterioles contains, or the, if you see the structure of the arteriole, it has endothelium and few layers of smooth muscles in the tunica media. But meta-arterioles are the blood vessels between the capillary and the arteriole. Here the tunica media contains smooth muscle, but they are very few in number. In the capillaries, tunica media is absent. It is lined only by the endothelial cells. In the meta-arteriole, these smooth muscles acts as a sphincter, and they regulate the blood flow through the capillary bed. So the meta-arterioles divides into capillaries, but there may be direct continuation between the arteriole and the capillaries also. The capillaries join to form the venules. Venules are also lined by endothelium. Initially, there are discontinuous muscles in the tunica media. Then the larger veins contains muscles in the tunica media. After the capillaries comes the venules. The post-capillary venules possess endothelial lining, basal lamina, and special types of cells. Those are called pericytes, which is also present in the capillaries. You already know the capillaries are lined by endothelial cells, and endothelial cells lies on the basement membrane. The basement membrane splits to enclose another type of cell called the pericytes. The pericytes are special types of cells which contains contractile elements in the cell like muscle fibers, actin and myosins. The function of the pericytes are they help in the regeneration of the capillaries or the venules. The pericytes are special types of cells which is present in the capillaries and the beginning of the venules or post-capillary venules. These venules are the principal sites for vasoactive agents such as histamine, serotonin, and they allow inflammatory substances to come out from the vascular lumen, and inflammatory cells also can come out through these gaps in the venules. They take part in inflammation and allergic reactions, migration of leukocytes and fluids. There is special type of endothelium are there in the venules of the lymph nodes. Those are called high endothelial venules. They are actually cuboidal epithelium. Those venules are lined by cuboidal epithelium. That's why they are called high endothelial venules. The bigger veins contains valves because the venous blood has to flow against the gravity and the venous pressure is very low. So there are valves in the veins of the limbs, especially lower limb, and they open in one direction from the lower part to the higher part. They don't open on the opposite side. If because of the inflammation on some other condition, if it opens, then it produces dilatation of those veins, and those conditions are called varicose veins, varicosity. So these valves become incompetent, and blood flow or blood is staggered in the lower limbs, producing dilatation of these veins, and these conditions are called varicose veins. The medium size, after the small veins comes the medium sized veins. The medium sized veins has all three layers, that is tunica intima, tunica media, tunica adventitia. Tunica intima contains the endothelium. There may or may not be internal elastic lamina. Tunica media contains smooth muscles, thin layer of smooth muscle, and tunica adventitia contains uh, collagen and other connective tissues. The large veins, the lumen of the large veins are very big, and it has also three layers: tunica intima, media, and tunica adventitia. The tunica intima contains the endothelium, some endothelial connective tissue, and internal elastic lamina. The media contains smooth muscles, but very few layers of smooth muscles are there. Adventitia is very thick and contains connective tissue. These are the pictures of the large veins and the medium veins. This is the large vein, maybe inferior vena cava or superior vena cava. Outside the tunica adventitia, there may be longitudinal skeletal muscles also in case of the large veins. This is a picture showing the arteriole containing 2 to 8 layers of smooth muscles and some amount of internal elastic lamina. In comparison, there is a lumen. This is a small vein. The lumen is irregular. Here, in case of the arteriole, the lumen is regular, and the tunica media contains very thin layer of muscle fiber. Here is an example of a lymph vessel, bigger lymph vessel. This looks like space containing some fluid, but there is no RVC. The lymph vessels starts in between the cells as blind canals or canaliculae. Those are the lymph capillaries. They join to form bigger lymph vessels. The lymph vessels has all three layers, that is tunica intima, tunica media and tunica adventitia. The tunica media contain only one or two layer of muscle fibers in case of medium size lymph vessels. Here is one of the lymph vessels, irregular lumen, and this is stained with spatial stain. This is the picture of the thoracic duct. All three layers are visible: tunica intima, tunica media containing smooth muscle, and the tunica adventitia. The lymph vessel contains multiple valves, and in between the valves, because of the wall is very thin, because of the collection of the lymph, it shows a beaded appearance. If you have noticed the thoracic duct, it gives a beaded appearance, and wall is very thin in comparison to the arteries and the veins. So in summary, large arteries, the elastic artery diameter is more than 10 millimeter. It has in the tunica media endothelium, connective tissue, smooth muscle. In the tunica media, there are smooth muscle and elastic fibers, and in the tunica adventitia, there is connective tissue, elastic fibers, and it is thinner than tunica media. The medium-sized artery or muscular artery, 2 to 10 millimeter in diameter, it has endothelium, subendothelial connective tissue, which contains some amount of smooth muscle and prominent internal elastic membrane. In the tunica media, there are smooth muscle, collagen fibers, relatively little elastic fibers. And tunica adventitia contains connective tissue, some elastic fibers, which are thinner than tunica media. Small artery, 0.1 to 2 millimeter in diameter, it has endothelium, subendothelial connective tissue, smooth muscle, internal elastic lamina. The smooth muscle layer is 8 to 10 cell layers, and outside there are collagen fibers. The tunica adventitia contains connective tissue, some elastic fibers, and it is thinner than tunica media. Arteriole, 10 to 100 micrometer in diameter, it has endothelium, connective tissue, and some amount of smooth muscle. The elastic lamina is absent, which is present in the small artery. See, in the arteriole, there are hardly one or two layers of smooth muscle, and the wall is tunica adventitia is very ill-defined. There may or may not be some connective tissue. In the capillary, diameter is 4 to 10 micrometer. It has only endothelium and pericytes. There is no tunica media, or there is no smooth muscle, there is no tunica adventitia. In the veins, there are large veins, medium veins, small veins, muscular venule, and post-capillary venule. The post-capillary venule is 10 to 50 micrometer in diameter. It has endothelium and pericytes. The tunica media, there is no muscle, and tunica adventitia is not there. So it has only endothelium, just like capillaries. Muscular venule is 50 to 100 micrometer in diameter. It has endothelium. It has smooth muscle 1 to 2 layers, and it has connective tissue, small amount of elastic fibers. It is thicker than tunica adventitia is thicker than tunica media. To differentiate between arteriole and uh, muscular venule, the differentiating point is the lumen. The lumen is larger in case of the muscular venule. In case of the arteriole, the lumen is smaller. Small veins, 0.1 to 1 millimeter in diameter. It has endothelium, connective tissue, and smooth muscles are there in the tunica media, 2 to 3 layers, which is continuous with the tunica intima, and it has connective tissue in the tunica adventitia. Some amount of elastic fibers are there. It is thicker than tunica media. In case of the veins, the tunica adventitia is thicker than the tunica media. The medium veins, 1 to 10 millimeter in diameter, and it has endothelium, subendothelial connective tissue. It has smooth muscles in the tunica media. It has internal elastic lamina in some cases. Tunica media contains smooth muscle. Tunica adventitia contains connective tissue, elastic fibers, and again it is thicker than tunica media. The larger veins, the vena cava, more than 10 millimeter in diameter, it has endothelium, subendothelial connective tissue, and smooth muscles in the tunica intima. Tunica media contains smooth muscle 2 to 15 layers, and cardiac muscles near the heart. Sometimes skeletal muscles are there, and the tunica adventitia contains connective tissues, uh, elastic fibers, and is thicker than tunica media. So this is one of the picture of the lymph vessels. It contains endothelium, and there are gaps or cytoplasmic flaps through which the macromolecules enters into the lumen. After that, it forms lymph, uh, bigger lymph capillaries, and these are the cross sections of the lymph capillaries. See the lymph capillaries, irregular in outline, bigger than the arteriole or the venule. Thank you.